Hi everyone, welcome to Enzymology classes. Today we'll be looking at another method of regulation of enzyme activity, and that method is through isoenzymes or isozymes. So, isozymes are the different forms of the same enzyme. So, they catalyze the same reaction, they have almost similar 3D structure, bind to the same substrate, but they vary in the kinetic property. So the rate of the reaction they catalyze are different for different isoenzymes. So they are having different amino acid sequences yet catalyze the same reaction. So these are the enzymes which display different kinetic parameters and different regulatory properties, as we will be looking at with example. So they are encoded by genes localized at different genetic loci. So they originate through gene duplication and divergence. So they differ from allozymes in that the allozymes are originated from the allelic variation of the gene. So they are originated from the mutation of the same gene that is localized at the same genetic loci, whereas the isozymes are localized at different genetic loci. So they exhibit differential electrophoretic mobility. So they are identified by their one of the most important characteristic, electrophoretic mobility. So as their amino acid sequence is different, they differ in charge and isoelectric point. So the researchers take this advantage to separate them on through electrophoresis. So what is the use of these isoenzymes? So they permit the fine tuning of metabolism to meet particular needs of a given tissue or specific developmental stage. So isoenzymes are the different forms of enzyme, same enzyme, which are expressed differentially at different tissue. So depending on the need of the tissue, the type of isoenzyme is expressed, so which meets the specific metabolic requirement of the tissue, and the catalyze reaction whose reaction rate will be optimal for the optimal metabolism of the cell. So similarly, some isoenzymes are specific for specific developmental stages. So when an embryo develops from a single cell zygote to the adult, some enzymes they synthesize various isoforms. So some of the isoforms are active in the early stage of life and others are active in the adult stage, depending on the metabolic requirement of the body. So as an example, we'll look at a very well studied enzyme, lactate dehydrogenase, which have different isoenzymes. So lactate dehydrogenase facilitates or catalyzes this reaction. You can see here. So they are mostly involved in anaerobic oxidation of glucose. So they convert from lactate to pyruvate and vice versa. So they are mostly involved in anaerobic metabolism of glucose and gluconeogenetic processes. So in human or in vertebrates, so there are 5 different isoforms of this enzyme. So actually this enzyme is they are the tetramers consisting of two different types of polypeptides. So one type is H type and the other one is M type polypeptide. So H, as the name indicates, it is mostly expressed, it is highly expressed in heart. So to remember easily, H stands for heart and M stands for skeletal muscle. So M type is mostly expressed in skeletal muscle. So as we know that skeletal muscles, when they contract vigorously, they are functioning mostly in an environment where oxygen is lacking. So M isoform is mostly involved in anaerobic metabolism of glucose, and also it is not inhibited by pyruvate. So in contrast, this aerobic isoform, which is present in heart, is inhibited by pyruvate. So these H and M isoforms, they are 75% identical in their amino acid sequence. So this percentage of amino acid similarity is an indicator of how much identical these proteins are. So they are highly identical. So they are also identical in their 3D structure and the other forms. The protein is tetrameric, as we have mentioned before, and so there are 5 different combinations of these H and M which are possible. So when all subunits are H type, it is H4, which is highly expressed in heart, and it has highest substrate affinity. And so substrate affinity is measured in their Km. So in other words, it has lowest Km, and when this number of H type peptides decrease and M type peptides increase, so finally when all of the peptides are M type, it become the muscle isoform, I mean the isoform which is found in muscle, and it has lowest substrate affinity indicated by a higher Km value. So here we can just claim that the Km for H type is lower than the Km of M type. So these are the properties through which their affinity for substrate is determined. So now we will see what are the advantages of these two types. So these types, the 5 different combination of H and M type of peptides creates different isoforms of lactate dehydrogenase. So when all isoforms are of H type, so it is LDH1, and this LDH1 is found mostly in adult heart tissue. So these combinations are taken from the adult rat heart tissue. So here, just to show that when the metabolism is changing from an anaerobic environment to a more aerobic form, the combination of these two peptides, they gradually change. So the adult is the most aerobic form, which has this LDH1 type. So LDH1 is having all H type of peptides, and similarly LDH2 is found in the rats having so who have developed from 12 days to adult. So they have 3 H forms and 1 M form. Similarly, LDH3, which has 2 M forms and 2 H forms, are found in rat who have from one day before the birth up to 21 days. And so this HM3 type, which is LDH4, is having 3 H isoforms and 1 L isoform, which is found from 5 days before birth to 1 day before birth. The M type, so LDH5, which has all M type of peptides, is found before 9 days of birth. So here we can see that the H isoform, which is more adapted to an aerobic metabolism, is increased in the heart muscle of the rat. So this tissue specific change in LDH isoforms helps the organism to survive in 2 different conditions, from non-aerobic condition to aerobic condition. Now we'll see how the tissue specific expression of the of different tissue of adult rat has different expression of these enzymes. So here this thickness of these bands so that indicate their level of expression. So here we can see that the heart muscles has very less H form and highest M form in adult rat. So this can be explained by assuming that so this M form, which is very less efficient in heart muscle, has to express to a higher level in order to perform its activity. So similarly, we can compare different tissues, and in muscle the condition is reversed. So here H type is expressed highest because it is the least efficient in muscle cell, and similarly this LDH5 isoform, which is expressed less in muscle cell because it is having highest activity in muscle. So similar is the case for liver cell because it is exposed to oxygen in adult tissue, and this H4 isoform or LDH1 is expressed to highest level in liver, and this M4 is least expressed in the liver cell. So this is in a nutshell the isoenzymes and the expression of these isoenzymes in different tissue of the same organism or at different developmental stages of one organism helps them to survive to different rates of metabolism.